Continuing along on motor neurons, we were talking about the cell body, the axon, and the dendrites. So we continue on here with the dendrites. Dendrites increase the surface area of neurons. They're allowed to make lots of different connections, as I showed you in the previous video. So if we increase that surface area of these neurons, what for? What's the purpose of that? Well, it can communicate with multiple neurons. And we've also learned that the actual dendrites have these specific enlargements called dendritic spines. I'm going to read this to you. Dendritic spines can function independently, sending messages back and forth to other neurons. The spine connects to an axon of another neuron. So interesting that not only are these dendrites receiving information into the soma and then redirecting it down the axon, but also there's a communication between these spines on the dendrite themselves. Let me show you. If this were the soma, here's the axon. We're thinking that information comes in through the dendrites, all these dendrites, feeds it into the cell body, and then transfers it down the axon. But now we know that on top of that, there are these specific spines on the dendrites that are able to communicate with the spines of another neighboring dendrite and send information directly. So that's a whole new ball of wax here. This is showing a presynaptic axon, so an axon from another cell that could be then communicating directly to these dendritic spines. Not just then, not just to the dendrite that then has to transfer to the cell body and down to the axon, but it's able to communicate at that level here and send or receive an information, which is interesting. They can change shape and size and number. So if they're observed in an individual, you can see that over time there's a change in what those dendritic spines look like. And why? So we believe that they're associated with learning and memory. And so when you train your brain to do certain things, you are creating more of these dendritic spines that make more connections with other neurons, and that's probably what learning is all about. They've seen that in lab animals, when they enrich the environment, it increases this dendritic branching, and it also increases the number. So you're essentially increasing the connections between neurons and allowing this mouse to learn. Very cool. So that's what you're doing when you're learning physio. You're increasing your dendritic spines. Skill training also has been shown to lead to the formation of new spines, and also it actually gets rid of older spines. So that often we think that a new skill sort of involves the rewiring of a circuit. And you would no longer have use for other circuits that have sort of been superseded by the new learning. Very, very crazy. Okay, those are dendrites and dendritic spines. Let's go to axons. So we think of the axon here in yellow. You've received that information, and either it's connecting with the other dendritic spines or receiving that information into the cell body and transferring it down the axon. You only have one axon per cell, but these can be amazingly long. They can be up to 1 meter long. So you could have a cell body in your spine and an axon that's coming from your lower limb. So it's interesting also to think of, how does a cell do this? Because these are microscopic cells, and yet it can be 1 meter long. It's got to be hard for the metabolism is all happening here in the cell body of the cell. and it's making ATP and it's making proteins. How do they even get to the other end, right? When it's so long. So we're going to look at that. How does, for example, an enzyme that's manufactured in the cell body actually travel to the other end? Let's say it's needed here in the terminal axons, in the terminal portion of the axon. How does it even travel there? So those are called axonal transport systems. Put a ruler in for you to show you that there is slow axonal transport, where material is going to move at about 2.5 millimeters a day. Right, so these are the millimeters here. And it's only about 2.5 millimeters a day. So very slow that it's going to go from that cell body to the other tip, to the end of the axon. It turns out that it does it in bursts of movements. It sort of pushes and then stops and pushes and stops. Okay, that's one form of axonal transport. Fast axonal transport is where it's going to move about 400 millimeters. So here, this is 10, 20, 30, 40, 50, 60 millimeters, and it's moving at about 400 millimeters a day. This is for synaptic vesicles and mitochondria. Mitochondria are going to also be transported so that they can make ATP for that end portion of the cell. A synaptic vesicle is the one that's going to release neurotransmitter. And if it's going to release neurotransmitter, it needs to be released at that very end of the axon. Let's make one here. And I'm going to have him be super long axon. And this is where the neurotransmitter is going to be. But it's not made there. It's made over here. It's made in the cell body. So if the cell body is making these, and then it needs to transport it through fast axonal transport all the way over to the terminal portion of this axon. I call it mass internal transit, because you make it, send it, release it, and then some of it is recycled and returns back to the cell body. But don't worry about that. Just think of it as being manufactured here in the cell body and traveling to the end of this axon that can be very, very long. More vocabulary when it comes to these neurons is, what is the axon hillock? What's the axon terminal? I've been using that word. The axon hillock is a location where the action potential is going to originate. So I'll make a drawing of that a little bit. And the axon terminal is where it's going to form a synapse with the next cell, which is the one that I just drew for you. So let's do it with this diagram here. If this is a cell body, it's going to receive an incoming message, correct? It's going to come over to the cell body, then it's going to leave along the axon and communicate with these other three cells here. Well, this section right here, this section here, which is the beginning of the axon, is called the axon hillock. And that is a location where it's going to send a message down the axon. If it doesn't get to the axon hillock, if let's say it dissipates and it ends up being just here, it's not going to fire. But if it gets to this location here, I'm going to make the message coming down. And if the message gets small and never reaches, nothing happens. If this message, oh my mouse, if this message reaches the axon hillock, it will fire down the axon. Okay, so I'm going to go back. The axon hillock then is that site where the action potential will originate. And the axon terminal then forms the synapse with the dendrites of other cells, and it contains synaptic vesicles. Let's go back. So the synaptic vesicles are going to be here, going to be released here. And these are all axon terminals. You can see here, axon terminals, And I'm going to make more neurotransmitter as it's released into that area, and that's how it's going to communicate with the next cell. Okay. I scribbled there. The wrapper. The wrapper is the myelin sheath of the axon. Let's just take this. I thought I could erase that, but I don't think I can. Let's see. Pen color eraser. Here we go. The myelin sheath is a cell, if you remember from anatomy, that wraps around that axon and actually increases the speed of conduction, makes it go faster. It's an insulator. It doesn't let ions leak in or out without you wanting that to happen. And this myelin then is formed from membranes of the Schwann cells or oligodendrocytes in the CNS. This you should remember from anatomy. Two types of cells that are going to just wrap around these axons and speed up the conduction and form insulation. There are spaces. It doesn't cover the entire axon. And those spaces between where one cell is wrapped around and not wrapped around, where there's no myelin sheath, is called a node. And it's the node of Ranvier. Maybe I'll draw that for you. So if you have an axon like this, and you have the cell body, there, my mouse just keeps reacting. And then the wrapper is going to go like this, and it's going to go like that, and it's going to wrap around, and this is all myelin sheath, but in between this region here and this region here, space that does not have the myelin is called a node of Ranvier. The next section we're going to talk about is the resting membrane potential and the action potential. So I'm going to stop this mini video now and continue on in part 3. Thanks for listening.